Good morning, class. In my previous class, we had discussed the naming of organisms and the taxonomical hierarchy, placing the organisms into different groups, ranking them one above the other. Today, I will start with some important definitions. Now, coming to these, first is taxonomy. Taxonomy is one of the oldest branches of biological sciences that deals with the naming of organisms, that is nomenclature, and placing them in groups or categories according to a particular plan or sequence, which means that organisms have been grouped in categories, names have been given to them, and this grouping is based on a particular plan. Then we come to numerical taxonomy. Numerical taxonomy, it aims to develop methods that are objective, explicit and repeatable, both in evaluation of taxonomic relations and in the erection of data. That means the information that has been collected based on relations, putting them in groups according to certain plans and sequences. That data that is collected can be evaluated keeping in mind the relations that exist between the different groups, different categories of living organisms. Then the next is cytotaxonomy. Taxonomy, now you have understood, is grouping of organisms in specific categories based on sequences. So here the category that is taken into consideration is cytology, cell structure of the living organisms and the genetics of the living organisms. That is cytotaxonomy. It is the information that is available from cytology. Cytology means the study of cell and genetics of organisms, which is needed for classification. Then the next is chemotaxonomy. Chemo word comes from chemical. So when we are taking into consideration the chemistry of the living organisms and how that is helping in classification of organisms in different categories. So chemotaxonomy is the application of chemistry of organisms for the purpose of classification of organisms. Then the next broader term that is used is systematics, which is a combination of nomenclature and the relationships that exist, the evolutionary relationships and how they can be compared. So systematics is the scientific study of the kinds and diversity of living organisms and existing relationships among them and the comparative and evolutionary relationships. As I told you, organisms, species is the basic unit of classification, the smallest unit of classification, and a species describes the living organism and talks of the relationship between different organisms and how based on these evolutionary relationships, the next category, they are grouped into higher categories like species. Many species form a genera, many genera, they form a family, many families form an order, many orders form a class, many classes form a kingdom. So this nomenclature, taxonomy and comparative relationships in terms of evolution, all that taken together, that is a broader term which is called systematics. So these are some of the very important definitions. You will have to learn them. Now we will come to the next topic, that is three systems of classification. These three systems of classification are the artificial system of classification, the natural system of classification and the phylogenetic system of classification. I hope all of you have copied these definitions. The definitions are given in the book also, but I have written here in an organized manner. So I will wait, copy, and then I will do the next. Okay, now we will move on to the next topic, that is 3 systems of classification. As I just now told you, first system is the artificial system of classification. Second is the natural system of classification, and the third is the phylogenetic system of classification. So 3 systems of classification. 1. Artificial system. 2. Natural system. And the third is phylogenetic system. These are the 3 broad systems which are used for classification. Now, we will do comparative study for these 2, artificial and natural. Artificial system of classification is based on a few characters. This is based on a few characters. Whereas the natural system is based on a large number of characters. The artificial system takes into account only superficial resemblances like presence or absence of wings, whereas the natural system takes into account important relationships between organisms based on DNA and proteins. Then the artificial system did not reflect any natural relationships among organisms. The natural system reflected evolutionary significance. This system, the natural system, reflected evolutionary significance among organisms. Then the artificial system led to heterogeneous assembly of unrelated groups. On the contrary, the natural system led to homogeneous means similar, homogeneous assembly of related groups. Now, keeping these characters in mind, you can see this is a very arbitrary system. So, it is an arbitrary system, and because of this, There was no proper way of categorizing organism into groups, so it was discarded. It was not accepted. This was a logical system. This system is logical because it has a basis of categorization, and so it is still accepted. It is still accepted. Now, in the artificial system, the categories that were made, did not tell us, did not convey much about the organisms, because there was no proper logical order in which this was done. Made a mistake in the numbering, so this was the 6th point. Then in the natural system, the taxonomic category of organisms tells us a lot, gives us a lot of information, gives us a lot of information about the organism. So here we have done 6 points where we have studied about these two systems, and at the same time we have also done comparison between the two systems. Now we will move on to the third system, the phylogenetic system of classification. Now the third system, the phylogenetic system, is based on the evolutionary history of particular group of organisms, which is called phylogeny. So this system of classification is basically based on evolutionary relationships, so it is also called evolutionary classification. Now species is the smallest unit. We have been discussing this fact that species is the basic unit of classification, and several species are grouped in one genera. So these species have a lot of resemblances with each other because they are believed to have evolved from a common ancestor, and that is why they have similarities and they are placed in the same group. So this is the third system, the phylogenetic system of classification. After this, we will do the 3-domain system of classification. Now, under 3-domain system of classification, domain is a category that is placed above the kingdom, and 3-domain system of classification. This was given by scientist, biologist Carl Woese and his associates in the year 1978. Carl Woese and his associates gave the 3-domain system of classification in 1978. So, as I told you, domain is a category that is higher than the kingdom. Category higher than the kingdom. So these 3 domains are, first is archaea, second is eubacteria, and third domain is eukarya. Now archaea, these are the oldest group of organisms. These are the true bacteria. Under eubacteria, we have the true bacteria, and eukarya are all eukaryotic organisms. That is, all those that have a true nucleus. These two, archaea and eubacteria, do not have a true nucleus. They are prokaryotic. These are the very primitive forms, and these are the true bacteria. They differ. They are different from each other. Though they are prokaryotic, but there is a major difference. So, archaea live in extreme environments. And eubacteria are found in almost all ecosystems. Found everywhere in all ecosystems. And these archaea, when I say they live in extreme environments, they are divided into three main categories. Methanogens, that are methane makers, and second category of these are halophiles, that is, they live in high salt concentration places, they are salt lovers. And third category of these are thermoacidophiles, that is, thermo means temperature, that is, they are both heat and cold lovers. Will be found in extreme temperature ranges. Now, the cell wall of all these three categories, that is, in general, the archaea is without peptidoglycan. They are without peptidoglycan. Now, the cell wall of eubacteria contain peptidoglycan. These are sensitive to antibodies. Sensitive to antibiotics. These are sensitive to antibiotics, and their cell membrane structure is made up of protein and lipids. And the true bacteria, they lack membrane bound organelles. The subgroups of eubacteria are cyanobacteria, which are the blue-green algae, mycoplasma, then gram-positive and gram-negative bacteria fall under this category, gram-positive and gram-negative bacteria. Then spirochetes, actinomycetes, and rickettsia. They all come under eubacteria. Now, eukarya, as I told you, all those organisms that have a true nucleus, that is a membrane bound nucleus. The nucleus is surrounded with a nuclear membrane. Now, the cell wall in these cases is also without peptidoglycan. Now, the animals, they lack a cell wall. All the animal cells lack a cell wall. They do not have a cell wall, whereas plants possess a cell wall. Plants possess cell wall. Then the cell membrane of all the eukarya organisms belonging to eukarya is trilamellar, made up of protein, lipid and protein. So this is a trilamellar cell membrane. Now these are resistant to antibiotics. These are resistant to antibiotics. Now the major groups that fall under this are protista, fungi, plantae and animalia. These are the major groups that fall under eukarya. In this, the major groups are methanogens, halophiles, thermoacidophiles, which live in extreme environments. These are found in all kinds of ecosystems and all kinds of environments, and they have been divided into major groups like cyanobacteria, actinomycetes, mycoplasmas, gram-positive, gram-negative bacteria, spirochetes and rickettsia. So this is the 3-domain classification system. And I have described the major things to you. Now we will move to each of these separately and will discuss these in detail. So first we begin with domain archaea. As I told you, they are divided into 3 main groups. Methanogens. Methanogens are the methane makers. Now these are found in swampy regions, in marshes, in the gut of ruminants. And when I say gut of ruminants, that is the buffaloes and cows, and also in the gut of termites. They produce methane from carbon dioxide and acetic acid. And the important methanogens are methanobacterium, and methanococcus. These are the examples of methanogens. Then we come to the second group, the halophiles. As told earlier to you that they are salt lovers found in high concentration of salts in oceans, etc. And they are aerobic heterotrophs. These are anaerobic autotrophs. The methanogens are anaerobic autotrophs, whereas halophiles are aerobic heterotrophs. And they have the pigment bacteriorhodopsin, which is purple, and they harness the solar energy from strong sunlight. And examples of these are halobacterium and halococcus. Then the third group, this was the second group, this was the first group. The third group are the thermoacidophiles, which can tolerate a lot of heat. They can be found in regions ranging 80 degrees Celsius to 100 degrees Celsius temperature, found in hot springs. These the term thermoacidophiles means heat cold lovers. Now they can be aerobic or anaerobic chemoautotrophs, and they are adapted to life above 60 degrees Celsius, generally found at temperatures 80 to 100 degrees Celsius. Even they can be found in freezing temperatures, 0 and below 0 degrees Celsius. So they are heat as well as cold lovers. They are found in hot sulfur springs, in polar ice cap regions, in geothermal where there is excessive heat. So both in extreme heat and cold conditions, and examples of these are sulfolobus, which is aerobic, and the anaerobic form is, this is the anaerobic form. I told you they are aerobes and anaerobes. So the anaerobic form is D-sulfuromonas. These are examples of the anaerobic thermoacidophiles. Now, these were about the three categories of archaea.